the camera. Just scoot to the left. Okay. Does anyone? We have a couple more chairs. Who wants the chair? Come on.
You're like a little in it. I like.
You'll be on camera.
I think that's probably good.
Okay. Amazing. Amazing. Okay. Thank you guys all for coming through. Justin, make sure that it's you. Thank you guys all for coming out. I really appreciate it. Who was here from last night also, just out of curiosity? Oh, wow. A lot of people here. Do you guys want to come up? Come sit. Come sit. Come on up. Yeah. Come on. Yeah. You got you. Come. We got you.
We are on the sidebar. That'll go.
Okay, so thank you guys all for coming out, and glad to see a lot of repeat faces from both tonight and tomorrow night. So that's very cool. And we'll just get started first. If you guys are on, and can you guys see? Okay, if you guys are on, follow us on Twitter and Instagram. We just got our Twitter account sort of back quasi, so you can take a picture of that and follow. Who's following us on Twitter? Okay, so all the people that don't have your hands up, give us a follow. And all the people who are not following us on Instagram should give us a follow. We post pictures. We have this amazing photographer here, so everyone give a round of applause to the photographer. And he's going to post all his pictures there, so if you want to see you can be there. Then we want to give a shout out to all the people that made this possible. First, Solana Foundation. Are you guys here? Do you want to say something for 2 seconds? Yeah, jump in.
Hey guys, I'm Colleen. I'm part of the team at the Solana Foundation. I do growth stuff. And one of the things actually, so like we're always looking for, maybe I'll step back. So the Solana Foundation, for those of you guys who don't know, like I think an analogy that I'll use to describe it is we think of Solana sort of loosely as this digital city-state. And what we want is for it to be like a flourishing digital city-state. And my sort of personal goal is for it to become sort of this digital city-state that has New York City level density of talent and capital and Tokyo level infrastructure. And like talent and capital, like that's a very broad set of things that can mean. Today in crypto, and certainly Solana is no exception to this, like one of the dominant use cases is just like speculative activity, which is fine and good. Like I'm sure there are people here who punch shitcoins, and that's great. To me, like that has product market fit. We don't worry about that so much. That's just going to take on a life of its own. What we're really focused on at the foundation is figuring out, okay, what can crypto enable that's, valuable for reasons other than speculation? And especially for those things that don't have product market fit, what can the foundation do to support that? So I, for example, have spent a bunch of time the last 2-3 years working with DePIN, so decentralized physical infrastructure. You guys might be familiar with projects like Helium that are actually building like real world infrastructure using crypto incentives. That's something we think is important because it's like a very tangible reason why crypto might be good. And DeSci, I think, is like sort of in the early cusps of demonstrating that as well. And so I got to meet Michael, I think maybe a couple weeks before Thanksgiving. And it's like, okay, I know very little about DeSci. I'm not a scientist by trade. But I can sort of see some of the opportunity here. And I've also been sort of tangentially associated with or exposed to like the process of science and some of the ways in which it's broken. And so just excited to learn, and thank you all for being here. Cool. If you have crypto related questions, feel free to come.
Okay, thank you, Solana. And then secondly, we have Stadium Science, and that's the company we started. Makes science a sport so that anyone can participate in it. And I think it's like one of the leaders in decentralized science now, so that is very cool. We have the Waterbear crypto community also supporting us here. So Waterbear is a meme coin token that is doing very well. And we're lucky to have their support to sponsor this talk. And lastly, without least, certainly not least, we have the Sci-Hub community. This is an incredibly powerful and ravenous community that is all across Twitter, and they're trying to free knowledge and decentralize knowledge. And they're both doing a great job. And we're going to talk a little bit at the end around like the role of meme coins in building community and in doing science, because I think science is sort of a community-driven thing, and meme coins are very good at capturing that. Okay, so if you want to get an airdrop from the Waterbear community, you can scan this and get the token address and fill it in, and I think we'll give you how many tokens with it? I don't know.
Sure.
So a certain number of tokens, some of they agree to give you. So this is part of the airdrop. You can get an airdrop from the Waterbear community. Oh, okay. People want free tokens. Okay, nice. Noted. Okay. Oh, okay. So what is DeSciNYC? I think a lot of you have been here before, but we've hosted Brian Johnson, we've had Michael Levin from Bioelectricity, we've had Martin Shkreli talk. We try and get people that 50% of the people love and 50% of the people hate. So we try and get people that are not doing normal sorts of science, that are trying to push the envelope in some way. And I think that's what's going to make decentralized science really powerful, is like not just getting people who are like, oh, I'm doing a PhD in like a new metabolic pathway or something like that, but people who are actually not people who have grand visions about, big next steps that we can take around like longevity or fertility or some of these big problems that are facing humanity now. So we hope to be the leader in that in the New York City area. And then, of course, we wouldn't be remiss without promoting our next event, which is with Suraj. And Suraj is amazing. I've known him for a long time, actually. He has like several million followers, I think, on YouTube. And he's an author of a book, and he's just like a very creative fellow, actually. Like, you've probably seen him on YouTube. He starts off every video with like, hi, I'm Suraj, or hello world, it's Suraj here. And he's going to be talking about AI and science. So he has access to ChatGPT 01, and I think he's trying to sort of figure out how you can do science with AI, right? So this is probably like what the future is going to be. Like the idea that humans do science is probably like in the same way that humans copyright or program or any of these things that will probably be like mechanized by AI. I think science is also very close to that happening there too. So he's doing some research in it and trying to invent new things with science. So I think this is going to be like a really, really good talk. You can RSVP with this QR code here. Okay. So what is decentralized science? So there's a lot of barriers in science today. Like it's really slow, and it's really inaccurate, and it's kind of shelled off. So like a lot of the research that's done today with like when like psychology is just like ends up being about like people that are currently undergrads at like a top tier university. So it's not generally applicable across a lot of different domains. So decentralized science is about like making science more accurate by like putting all of the data on the chain so you can have provenance around where the data comes from. It's about speeding it up and gamifying certain parts of it so that like it's not just about like one lab doing something. It's about people sort of competing for prizes, and if you're at the talk from Robin Hanson last night, sort of competing for prestige is another thing that can be sort of gamified and incentivized, as opposed to sort of papers taking like 4 years to publish or a PhD taking 6 years. It's also like about like expanding participation. So with like a traditional like a DeFi opened up financial institutions for a lot more people that may have been like unbanked and not able to participate in that. And it's the same thing with decentralized science. Like science is only accessible sort of to a small group of people, but really anyone who's sort of producing data or able to analyze data should really be able to participate in science and also to be incentivized to for the work that they do. And I think if you look at a lot of like academia, there's this guy on Instagram who's like, he's a professor at UCLA, and he's always complaining that he's not paid properly, blah, 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 which is probably true. And I think DeSci offers people sort of to be paid in correlation with how much actually good work they do. So they get paid and how difficult the problems that they solve are, or how experienced they are, or the value that they create. So DeSci tries to fix this. So as an example, we had this idea. We've been working on DeSci for a while, and we were like, okay, it's time to actually do a project. So we created this thing called Stadium Science, and the idea is to sort of gamify science so that it's actually like interesting and fun, and people can compete, and it's sort of like a social media thing. I think one of the things people have to think about when they're doing like science is like you have to entertain people. People have to like actually enjoy being there and enjoy participating and like thinking that they're like working towards some sort of goal. And like what types of things have a really great way of doing that is like a game. So imagine instead of just like doing science alone, you're doing sciences with like a group of people. So we came up with this idea of combining like social media with like science. So yeah, people are posting and creating data. So the way it works is like, how many people here have an Oura ring actually? I'm kind of curious. Okay, wait, I'm going to skip ahead to a slide because I want you all to fill out this form because I would love to talk with all of you and just put in your email address if you have an Oura ring. Actually, okay, yeah. So if you have an Oura ring, fill out this form and I'll send you an email later. We'll incentivize it for you. We want to get your feedback. But we're looking for beta users. And the way it works is you connect your Oura ring to the platform, and then you basically create like a betting market around sleep data. So people will participate in these leaderboards to see who can sleep the most, and then you can say like, okay, now I'm going to compete on sleep. But then you can also do cool things like you have insider information. Someone like knows that you're out partying really late last night, and you can short them or something. But then there's other things you can do.
You can throw a party.
So then you can see if you go to stadium.science, you can see all of my sleep data, and you can see if I took this from a day that I was actually sleeping very well. And you can see sort of, and all this data is put on the blockchain. It has provenance to me. So if you ever want to try and reproduce any sort of science for that, you can easily just look at the data on chain. And then the next question is okay, so you're collecting this data, but like data alone isn't really that interesting. So we created these things called like power-ups, or these are the interventions that work well. So I'm tracking all the things I'm taking. Maybe I'm taking caffeine, maybe I'm using a sleep mask. And these are sort of protocols that would be like interesting for people like in a science realm. Like, and you could say like, oh, if you take melatonin, that is a really, that thing actually helps me, or that thing doesn't help me. And so you can actually buy and sell. This is a pump.fun melatonin, and you can buy and sell melatonin tokens, and that will rise and lower with the price of melatonin, of how effective it is. So if people see that I'm taking melatonin and it's impacting my sleep score, that melatonin molecule has more value, and you can vape into it in the same way that you might think that like a Dogecoin or what's the guy, the sad guy or cool guy? Like cool guy has like mimetic value that can see melatonin actually has like a really good thing and make it useful across a lot of different domains. So you can like buy into that. And then the last part is you can do like prediction markets on it. So you can look at my sleep score, you can look at the drugs I'm taking, my physical activity, maybe I'm posting a lot on Twitter, maybe I'm trading a lot of meme coin stocks, and you can see that based on my wallet history, I'm not going to be sleeping well. So you can bet on my sleep being good or bad. So this is sort of the final thing. That allows people to sort of gamify and participate in science in like a more open way. So if you want to participate, we're still looking for users. So we have a we're looking to expand it out more as we get feedback. So if you want to participate, we'd love to talk. And then if you want to download the app, you can download the app for iOS or Android. You can stalk my sleep if you're interested. Okay. We'll give them time to get it. Thank you. Oh, lastly, we're hiring. So if you want to help build this, or you want to help with jobs, or you want to help organize DeSciNYC events, we're looking for fast people that are active. Business development, marketing, design. You can scan this QR code here. The important thing is you're fast, right? So like we're always looking to like iterate quickly on these things. So that's sort of the main thing we're looking for. Okay, I think that is all. So now we will talk about the water bear. We got this online. And this is probably why you guys are... Oh, actually, before we do that, can we get a picture of the audience? Come up and... Yeah. So, on 3, this is amazing photographer extraordinaire. Yeah. Okay, so on 3, say, do you guys can loop around maybe? I don't know. Yay! Okay, on 3, 1, 2, 3. Waterbear! Waterbears! Awesome. Okay, thank you guys all. Okay, so let's get on to the next portion of this, which is on the wonderful tardigrade here. So we will open this. Does anyone have any questions about decentralized science? Anyone? Or someone must have some.
So, are you guys presenting at JP Morgan this year?
No, we are not. We don't really do conferences, but maybe we should. If you have a connection, who said that? Who accepted it? Oh no, but maybe we should. Yeah. Well, I mean, come chat with me after, and we can chat. Anyone else around decentralized science or anything else? Speakers, if you want to speak, let me know. Yes. All right. Well, without further ado, we will welcome our main presenter for tonight. So give him a round of applause. He's wonderful. He's on DeSciNYC speaker or attendee, PhD student and postdoc and wonderful human. So the stage is yours.
So, thank you, Michael. I'm a scientist. Actually, disclaimer, I'm a neuroscientist. I don't do research specifically in tardigrades, but as a scientist, I've always been fascinated by them, these creatures and their resilience. And so I agreed to give a talk to help us all understand a bit better these creatures and the nature of their resilience. I do research mainly in neuroscience, neurodegeneration, psychiatric disorder. So now let's dig into the science of tardigrades. So first of all, what are tardigrades? They are small opportunistic organisms, basically humans. They're microscopic and invertebrates. They're typically between 0.3 and 0.5 millimeters in length, so very tiny. They were discovered in 1773 by German zoologist Johann Goethe, who named them kleine Wasserbeer, which means in German little water bear, the colloquial name in English. But then the Italian zoologist and biologist Lazzaro Spallanzani was like, you guys are amazing, you deserve a scientific name. So he gave them a Latin scientific name, tardigrada, from Latin for tardi, slow, and grada, gradi for step, because they move very slow, and they have a slow metabolism as well, which is to their advantage, as we will see. Everyone wants to speed up metabolism, often, to get thinner, but the slow metabolism is better for longevity. Who in the audience is a scientist? Who's in biological science? Okay, just to know. So, you can interrupt me also during the talk if you want, if you don't understand certain things, but I will be making it very easily accessible. So, let's get acquainted a bit more about these animals and where they're from. So first of all, they are animals. They're not bacteria or viruses. They're animals, which is important as we will see. So they pertain to the kingdom Animalia. They're multicellular organisms, like us in a way. Their phylum is unique. So they have their own category, tardigrada, which no other creature is part of, which is pretty peculiar. The superphylum is Ecdysozoa, which also includes arthropods and nematodes. And these guys have been around for many years, around 500 billion years. So they preceded dinosaurs, and they also overcame the 5 big mass extinctions on Earth. So they are survivors. Now the question is, why are we talking about tardigrades in first place? I can summarize some of it with these publications. So their resilience to extreme environmental conditions can inspire us in terms of biomedical research and achievements to understand how we can survive extreme environments and extreme conditions. So their motto is improvise, adapt, and overcome, something we should always learn in many different fields of life. So the peculiarity with tardigrades is that they can go over from a hydrated and active state over to this tone state in which they can resist pretty much anything. This state is called cryptobiosis, from the Greek words for cryptos, which means concealed, and biosis, life process. So they literally get into a concealed, hidden, they hide from threat. It is collectively called cryptobiosis, but then the specific tune state they get in depends on the threat. So if you're talking about too little oxygen, they go into anoxybiosis. If the temperatures get extreme, they go into cryobiosis, and so on and so forth. By getting into this state, they can resist temperatures up to minus 328 Fahrenheit for up to 20 months. They can resist temperatures up to 302 Fahrenheit. They even can withstand 6,000 atmospheres, which is a lot. Right now at sea level, we are under 1 atmosphere pressure, and humans can resist up to 6 atmospheres. After that, we die. Get crashed. And this guy is 6,000 atmospheres. This is more than in the Mariana Trench, which is, the deepest trench in the ocean. This is insane. They also can resist pure vacuum. So they can resist implosion forces when they're in the tune state. And also is extreme osmotic pressures, like when salt concentration in water gets too high and we will just desiccate, they can resist that too. So there's a lot we can learn from them. Now, if we look a bit closer at tardigrades, they are pretty simple. Like they have a mouth, they have a very simple brain and simple nervous system, they have their digestive apparatus, simple reproductive system. They're fucking simple. So what's their deal? How do they do this? What's their secret? To understand their secret, we first need to understand what is a hazard to life. Everything I mentioned before, like extreme temperatures, extreme pH, extreme pressures, dehydration, irradiation. Why is this a threat to us? Any suggestions regarding to any of these? But they all have a related hazard to our cells, to our organism.
DNA damage?
Huh?
DNA damage?
For one. What else?
Bodily damage?
Huh?
Just bodily damage, general?
Also, exactly. So, all these extreme conditions pose a hazard to our protein integrity, our lipid integrity, and genome integrity. So, our cells, animal cells, to survive, rely on the proper integrity of proteins, which basically make our cells function, lipids, which cover our cells and sort of seals it from the outside world, and our DNA, our genome. Proteins have a very specific three-dimensional structure. Like our cells are full of enzymes and other proteins that makes it alive, makes it work. And under extreme temperatures, they misfold, they don't work anymore. under extreme pH or pressures, they are all altered. Same thing for lipid. Our cell membranes are made of lipids, and if we go in extreme temperatures or pH or anything else, they get completely distorted. And lastly, and very importantly, DNA, our genome, what codes for our life processes. It's very sensitive to irradiation, to temperature, to pressure. So our cells, animal cells, are very sensitive to this. Why aren't tardigrades cells sensitive to this? And their cells are just like ours, they're animal cells, if we look at them from a biological perspective. So to understand how they are resilient to all these stressors, despite having cells similar to ours, I brought an example from a study that was published very recently on a new species of tardigrades. And their secret is really in their genome, in the genes they have. So, like us scientists always do, we get nosy and we go and peek into their genome, into their genes. Are you guys familiar with what RNA is?
Yes.
It's the intermediate from DNA. We produce RNA, which is then translated into functional proteins that make us work. So they sequenced their RNA of these new species, and they found 14,000 genes, of which 30% are unique to tardigrades, which is crazy. Even our genes have 70% homology with mice or other creatures. Like, we don't have these unique genes. Tardigrades have 30% unique genes to them. Then researchers kindly irradiated these guys with 3,000 to 5,000 gamma rays, which is 1,000 times more than what we would survive, to see what happens. So they found that upon irradiation, tardigrades upregulated the expression of 2,800 genes involved in DNA repair, cell division, and immune response. Now, one of the genes that caught their attention, amongst many, is this TRID1, which encodes for a protein that helps repair double-strand DNA break. You know, DNA is a double helix, and upon irradiation, it breaks. We do have proteins that help in repairing DNA, but they are not nearly as efficient and quick as theirs, like this one. They also express the same ones that we express, and additionally, they have their own unique ones that are very, very, very efficient. Additionally, they found that 0.5 to 3% of the tardigrade genes were acquired from other organisms. Thieves. They stole genes from other organisms to be able to survive better through a process called horizontal transfer. And this is interesting. What is horizontal transfer? Usually humans acquire their genes in a vertical fashion. Parents, offspring, parents, offspring. Some creatures are able to acquire genes from other species. Then this is already about gene transfer. Now, in their tomb state, these guys are a little bit permeable to DNA in their environment, from, for instance, bacteria and fungi in the soil, in the water. And as I just said, they have amazing DNA repair proteins. So these proteins can swiftly and efficiently put that genome they find in the environment into their own. A bit like a very efficient CRISPR-Cas system, if you've ever heard of the CRISPR-Cas genome editing system, they have it inbuilt. So they identified several proteins, like up to 3% of the proteins that they acquired from other organisms. One of these is the DOTA1, which seems to be acquired from bacteria, and this protein enables them to produce certain pigments that are very efficient antioxidants called betalains. And only these pigments can get rid of or stand up against 60 to 70% of the free radicals produced upon irradiation. So already just by acquiring this gene, they can cover the 70% of the radiation damage, which is pretty incredible. Another feature besides this resistance to irradiation, which is very important, guys, because Elon Musk wants to ship us all to Mars to know how to withstand radiation attack, because there's a lot. Another feature is their ability to survive in extremely cold temperatures. Now, what I put up here is an animal cell, could be your cell. It's a tardigrade cell. Do you guys know why we cannot freeze our cells? What happens to our cells if we put it in liquid nitrogen, you pre-freeze it?
Vitrification?
No, you wish.
Oh.
That would save you.
Oh, why you can't?
Why you can't?
Because of the ice cells.
Huh?
The ice cells. The ice crystals.
Right.
Yeah.
There is an ice crystal buildup in the cell that will pierce through all the organelles that keep your cells alive, would misfold all the proteins, and just basically destroy everything. Why do tardigrades don't build up crystals? Again,
They have no water.
Huh?
They have no water.
They do have water.
Oh.
They pump it out, but in the process of doing so, they need to protect their proteins, their lipids, and their DNA.
Huh.
So they evolved to produce specific proteins, such as LEA proteins, tardigrade specific intrinsically disordered proteins that can stabilize basically all their macromolecules inside the cell. Basically, proteins like LEA, they attach and bind to the proteins and make sure the three-dimensional structure is kept proper. And these all in the process of pumping out water and ions as the temperatures go below too much. Additionally, the TDPs, these tardigrade-specific intrinsically disordered proteins, they build up a gel in the absence of water that maintains all organelles and all proteins, DNA and lipids in a good conformation. These LEA proteins are actually found also in plant seeds. That's how seeds can be dry but alive and give them rise to life when they come across water again. Also, they produce the sugar trialose, and they also produce glycerol. You know glycerol?
Yeah. Antifreeze.
Exactly. They have their own antifreeze. They produce it. So in the process of getting into the tune form, they ensure that the water inside their cells don't get frozen. And this is the process of vitrification, when you avoid the water to freeze into a crystalline state. So they do have the time to pump out all the water and desiccate by the time the temperatures go far too low. And then glycerol and trialose together also provide an osmotic safety measure, because as you pump out all of the ions and water, you need to maintain an osmotic pressure inside the cell for it not to pump. And they also have that function. And also importantly, when these tardigrade-specific intrinsically disordered proteins form this gel, this slows down any molecular movement inside the cell, slowing down the metabolism almost to zero. One of our issues with longevity is the fact that we have a fast metabolism. So we produce free radicals, which then damage proteins, damage lipids, damage DNA, and we age. By slowing the metabolism, they can halt this process. It's like, you see how fast I can move in air? If I put you in a swimming pool full of dense gel, you would move much slower. It's the same thing with their molecules, their enzymes, their proteins, whatever, inside the cell is slowed down in a gel. And also they have very efficient antioxidant measures like superoxide dismutase, glutathione, peroxidase, and catalase are our gold standard antioxidant measures, and they have it too. We do have heat shock proteins ourselves to maintain structural stability of our proteins, but they are not nearly as efficient as the LEA proteins and the TDPs. Also, they have these DeSciNYC, which are damage suppressor proteins. They bind to DNA and protect it in a very efficient way. We wish we had something like this. That's how they survive irradiation in the first place and have the time to actually get into a tune state. So if we were, for instance, to transfect ourselves with CRISPR-Cas systems to make our cells express DeSciNYC, we would already be able to survive a bigger dose of radiation. And these are very specific to tardigrades. No other creature on Earth has them. So where are we at with cryopreservation, for instance? So we can cryopreserve human cells. I do it routinely in the lab. We can cryopreserve embryos. We use similar systems as the tardigrades. We can use trialose and glycerol to vitrify the cells, avoiding ice crystals. We can use serum albumin, which is a serum protein to shield the cell membranes, a bit like trialose and Lias does for the tardigrades. We can also cryopreserve the whole human. I don't know if you heard of this company called Alcor. I think they're based in California. They provide this service for a few $100,000, and they basically cryopreserve you at time of death. The only issue is that we have no idea how to wake them up, unlike tardigrades. So when tardigrades go into their tune state, they take 30 minutes to re-expand and revive into a normal hydrated state, which is not the case with cryopreserved humans. So there are some billionaires that get cryopreserved in the hope that in the future we'll find out a way to revive them. So there's a lot we can learn from tardigrades, especially these billionaires' hope. Now, in terms of potential future applications of what we know so far about tardigrades, well, their LIA proteins and TDPs can potentially help us maintain biological structure more, especially in industrial production. Because, for instance, liposomes, have you ever heard of liposomes? They're used in cosmetics, they're used in medical treatments to deliver drugs, but they're very hard to handle because they aggregate and their chemical structure is a bit messy. So with TDPs, we could stabilize liposomes and other biomaterial that we are producing for our own purposes. And also some research groups have actually transfected human cells in cell cultures in vitro with tardigrade proteins that should help against irradiation, and they showed that they actually got protected from gamma radiation and UV light. So we are still far from transfecting humans and altering their genome this way, but it could be a potential future way of shielding us, at least from some damage. Now, the last and very important question. Is resilience equal to longevity? What do you think?
Yeah.
Yes.
No?
Yes.
Yes?
Yeah.
Yes.
Longevity is just like you mess up and then you die. So maybe if you're more resilient, you'll never mess up and you won't die.
If your cells aren't damaged, right? I mean, like, that's what ends up killing you. Is that your mitochondria keeps getting more and more damaged and you can't reproduce your cells anymore.
Right. It's yes and no, and not necessarily, in the sense that the tune state, the halting of metabolism, doesn't mean rejuvenation. So any damage you accumulated by the time you go in tune state, it's going to stay there. So you're very resistant, but your aging process is not rejuvenated. So how long do you think tardigrades live?
2 years.
3 years.
Huh? 2 years?
A month?
6 months.
A month?
30 days.
6 months.
Yes.
Come on. That's a
Weeks? Okay, wait, but they can be in hydrated state and in the tune state. How long would they survive in hydrated state?
6 months.
5 minutes.
My God.
2 years.
So they can live up to 2 years in their hydrated state, which is kind of a lot for the size they are, and decades in their tune state. Hypothetically, even 100 years, but the longest recorded is decades. It's still long compared to other arthropods or nematodes in their superphyla, but it's not super long as one would expect, right? So resilience is not necessarily equal to longevity, and to have a comparison to help you understand this notion, there is a natural animal, an animal that can live forever. The planarian flatworm is not nearly as resilient as tardigrades, but they have an amazing regeneration capacity. They can live forever if they are not in a very, very threatful or harsh environment. Like if we keep them in a lab in good conditions, they literally can live forever. And their regeneration capacity is so huge that if we cut this worm into tiny pieces, each piece will give rise to a new, entirely functional individual. So planarians are studied for their rejuvenation and longevity, while tardigrades are studied for their resilience to extreme environments. So in a future in which we can alter our genome, we'll probably become half planarian, half tardigrades. So, with this, I end my talk, and I'm open to take questions, and hopefully I can answer them.
So a lot of the proteins that you mentioned are what we would call enzymes. So
Sorry?
a lot of the proteins that you're mentioning are basically fall into the class of enzymes. So it seems like they had a supercharged enzymatic system, so like their DNA helicase or
Right.
whatever checkpoints they're using between their messenger RNA and replication state there is supercharged versus the speed of their actual motion or activity. So how much do you happen to know, like how much of the focus on the research has been on enzymatic activity when comparing all of these different proteins? Because it sounds just like the first thing popping into my head is, okay, DNA repair is you have DNA checkpoints,
Right.
so but those are all enzymatic functions within a cell.
Right.
So
Good point. Very good point.
I'm just wondering if you had any information about like how much of it is enzymatic activity, like a supercharged enzyme system versus
They are
conservation
more
of energy.
right. Their enzymes, for instance, for DNA repair, are more efficient and more high fidelity than ours. But that's not the only trick. So they produce proteins that put DNA into a little bubble where enzymes can act quicker and faster. So they actually change the biochemistry of the nucleus. They build small droplets, these proteins, some proteins they produce, and those droplets make it easier for the enzymes to reach and repair with high efficacy. We don't do that.
Isolating the DNA while the enzymes do the repair.
Right. So
It's pretty
there are
dark.
proteins that get upregulated, and then you see the DNA where it's broken in a bubble, where enzymes can flow in quickly.
So it's like penocytosis fix and gone.
Precisely.
Yes. That's awesome. Thank you.
Yeah.
Why did the tardigrades evolve the ability to
What
be,
decentralized science is.
why did tardigrades evolve the ability to be so resilient? Where did it come from? Did they have to survive in space?
That's a good question. So it's not entirely understood. The potential option that they evolved like this to survive in space is actually very interesting. However, I think that if you look at the evolution of Earth, you had periods with very thin atmosphere, no ozone, a lot of irradiation. And you had also with these atmospheric changes, you had different pressures. You had life that had to go into the oceans, but then the pressure was an issue. So I think the fast evolving or the evolving situation on Earth pushed them to get that resilient. And the fact that they are so small and simple poses an advantage because, our complexity is a limit, especially when cryopreserving a human being.
So how quickly can they adapt? I'm thinking of 3-body problems, the book now series. But, the situation of these 3 suns creating different environments, so temperature, gravity, etc. Sometimes it can be slower, sometimes it can be like that. How quickly can they actually adapt to like an extreme of some sort?
That's a very good point. Got you. So they are not so quick. The process can start within minutes, but it takes about 24 hours. So on Earth, the conditions don't change that fast, right? So it's fine. But if you take a hydrated tardigrade and immediately place it in the vacuum of space, it might not have the time to withstand the implosion forces and the radiation and the temperature all together. If you bring up there a tune state tardigrade, yes, they'll survive fine. And also, not for too long. But yeah, so the time is important.
Going down.
Also, you talk about the 2 states of the tardigrades being from hydrated to the tomb state, and also that they're incredibly slow-moving creatures. How long does it take for them to get to each state? Like, let's say they're hydrated and they get to a tomb state. Like, what is the process of that? And have they been studied after having been in tomb state for an extended amount of time, being rehydrated? How does that also change the composition of the organism?
So, as I was saying, the transition to tomb state takes usually up to 24 hours, also depending on the threat. Are they being irradiated, or are they in a place without water or without oxygen? It induces different tomb states, slightly different. So it usually takes 24 hours, can take less. And then once you rehydrate them, once you put them back in a favorable environment, it takes literally just 30 minutes for them to be fine. And in longitudinal studies, they see that even after a year or so of tomb state, they're just doing great. You rehydrate them, they're perfectly fine, their composition is normal. So in this sense, they're very resilient.
Interesting. So it's not really a choice of the tardigrade to enter those states, it's an environmental reaction.
Yeah, it's a reaction to the environment.
I have another question, sorry.
Go ahead.
So I heard that they have a nervous system,
They do.
and I wonder how that is impacted by their environmental conditions. Because I just kind of figured that like maybe that their nervous system might have something to do with the sort of physical state that they get into. But
their nervous system is pretty simple and mostly involved in their movement. They respond to the environment through sensing vibration or chemical composition of the environment. So through various senses, they sense when it's time to go to. And also the cells all around their body, if they receive any too high radiation dosage, they decide, okay, the whole organism goes into tomb state. So it's not directly tied to their nervous system.
So on the perspective of us one day going into space, since it's quite impossible to transfect all human cells, could you think of any other method of biologically engineering humans, maybe across hundreds of years, to be able to develop the proteins that would give us the resilience that they have?
I think transfection would be the fastest way, but then you would need to transfect all of them.
And how realistic is that?
For now, not very. Also because we are too complex. You don't know what those TDPs will do to our brain cells or our lung cells. They might induce denaturation of our own proteins. We don't know. So it's very far-fetched.
How do they reproduce?
So they can undergo both sexual and asexual reproduction. Can be a male inserting the sperm into the female, or the female can undergo parthenogenesis, meaning their eggs just start dividing without having been fertilized, and they produce clones of themselves.
How does that lead to like evolutionary pressures and selection?
Right. It's in a way it's disadvantageous because you don't mix the genome, you don't get more variability. On the other hand, it allows the species to, go forth. And since they already evolved so many resilience mechanisms, they're like, you know what?
Do they have any like light sensors or any sort of visual system?
They have a very simple eye system. It's more visual cups.
Got it.
They can only detect light intensity, but not make up a clear image.
Do they use that at all for feeding or defense?
Yes.
Or
to understand their environment.
Got it.
Yeah.
So kind of in the context of their resilience in like a broader ecological sense, like what's the impact on, creature A eats a bunch of tardigrades and, tardigrades are now resistant to the acid. So kind of what happens then, like if I were to just drink an entire cup of these things, would there be adverse health effects?
Actually, good question. I have no idea. That's something that is
could make this sport.
That's your next paper.
Yeah.
Do they have a for somebody's PhD?
Do they have a predator? Yeah, we can find out. There's other arthropods and
Okay.
What about threat detection? Because you said there's different types of tomb states, depending on what the threat is. So there are unique ways of detecting what type of, like if it's already female creature or so.
Right.
Like do they have unique ways of detecting
Yes.
which specific threat
Yes, and that is not yet totally understood. Pressure, they have their cells, when they feel too much pressure from their cytoskeleton, these triggers all the gene expression needed. Also for temperature, when the temperature goes too high or too low, there are molecular changes in the cell. And this triggers an immediate gene expression of the genes that are needed to protect towards that state. Chemically, they have small fimbria and little microscopic hair to actually sense the chemical composition in terms of salts and osmosis to also warn them of the environment. But exactly how they sense everything is not yet characterized.
How do they derive those traits horizontally, you said? I suppose getting them from their parents.
When they're in their tomb state, they are somewhat permeable to
Got it. Okay.
environmental DNA. So it just flows in and their cells just insert something with their own. It can go south. It can be shitty DNA, gene they don't need. Like they wake up with a green it's south. I guess they'll die out in that case.
They go into like two different forms, or really just one of the six?
No, it's usually when it's horizontal, it just comes from other organisms in general.
Is the like there are a number of papers cited here. Is research into tardigrades, is it like a hot topic, or is it like a niche thing that only some people do? Like, how would you characterize it?
It's both. Meaning that it's a hot topic, but there are not many groups researching. Because it's a very, very specialized niche animal. So,
Yeah, how did you get into it? Because you're a neuroscientist, so
Right.
this is not that.
it's not that related in a way. However, they're intrinsically disordered proteins that keep proteins in such a stable state might be interesting for my research in Alzheimer's disease and Parkinson, because these neurodegenerative diseases depend on the fact that some proteins in our brain cells denaturate, misfold, and kill neurons. They form these beta amyloid plaques, and so there's something that goes wrong with our own protein stabilization system that we don't really understand. What if their protein stabilization system could help us keep things right so that we don't neurodegenerate? So in this sense, I was interested in how they do that.
Actually, one question just to flood this thread. I'm curious, like the story around this. Was this like your advisor suggested you look into tardigrades? You like stumbled upon it randomly on YouTube?
No, so when I studied university before my PhD, we studied them as in biology because they're so amazing. And then when I studied neuroscience and I learned about how we neurodegenerate, I kind of remember. So I read a bit more into that. And also, as a researcher, the more you research into our cells, the more it comes up to you like, hey, but those creatures, they don't suffer this, they're resilient to that. So how was it that they did that? So I was just reading here and there about them.
I have two questions. First, how big is like tardigrades?
0.3 to 0.5 millimeters.
Oh, so we cannot keep that pad.
You can, but you need a microscope to say hi.
Wait, on the question of reproduction, on what basis would a female's eggs start dividing? Like on how they can clone themselves? Like on what basis would that happen as opposed to just like waiting to be
fertilized?
Yeah.
Good question. I actually don't know if there is a time in which there's no male around. Let me do it myself.
Does their size play a role in their resilience? For example, if they were larger, would they perhaps be less resilient to certain species? So?
Yeah.
Hi. So your talk was very interesting. I similarly thought that, there's a lot of opportunities to leverage their proteins to actually help with a lot of the human protein misfolding conditions. Kind of like you said, Parkinson's disease and Alzheimer's. So I'm kind of wondering from that perspective, do you feel like augmenting their proteins to be able to cross the blood-brain barrier is going to be a more viable solution, or augmenting their proteins so we can actually have it cross the human's blood-brain barrier for therapeutic purposes, or gene therapy? Because I'm thinking with like amyloid beta plaques, right? That's a potentially an interesting area to explore where you can actually mitigate that misfolding issue.
Right. So I don't know if you've heard in the past year and 2 years, a lot of companies have released antibodies against beta amyloid plaques to have them cleared out, and that barely worked.
Yeah.
There was a big scandal that FDA approved those antibodies, but the results were shitty. So I guess just introducing proteins like that across the blood-brain barrier to stabilize amyloid beta wouldn't be the strategy.
Okay.
But having them being expressed by the neurons might be, because they misfold inside the neurons, and then they form the plaque, and then
fascinating.
Yeah.
Okay.
Agreeing with her, fascinating talk, and it's clear these guys have a supercharged mechanism for repair. I'm just wondering, do they have similar mechanisms as humans for like natural killer cells or that kappa B to come in when there is a mistake to eliminate that so it doesn't get replicated and become what we would call cancer, but for them could just completely fatal.
Right.
So do they have like a mediation system of sorts?
So in that sense, what they have works for them. They have a very rudimental immune system. They don't have all the specialized immune cells that we have. It works for them. They do have similar immune pathways that we had. Also, like phagocytic pathways and kappa B inflammatory pathways similar. Evolutionarily a bit more ancient. We wouldn't survive a sec with their immune system. So, there's always...
No, I mean, it's having the supercharged repair system is really pretty cool. But mistakes still happen, so I wonder, they could expect that.
No.
Fail safe.
They're more high fidelity in their repair than we are. Yeah.
No, that's very cool. Thank
They're
you.
more
Thank you
efficient.
so much.
All right, we'll do some rapid fire questions. So, who has a question? You go, and is there any two more? Okay, go.
Okay, mine is quick. What's the plant or animal equivalent of the tardigrade that's being studied right now for rejuvenation?
For rejuvenation, planaria, the worm I presented at the end, they have the most amazing rejuvenation capacity.
Yeah, go.
I have two short questions.
Rapid fire, yeah.
So, do we know which genes have been under positive or negative selections over their evolution? And yes.
That put pressure or relieve in their evolutionary.
Yeah.
For sure, the DSU, the damage suppressor proteins, they gave advantage, and the ones that didn't give advantage, I actually don't know because I looked only at the ones, my mistake, that gave advantage.
Okay. And the second question, do you have any idea about the molecular mechanism of how they can be resistant to the acidic environment?
Yes, the cuticle that they form in the tone state is of some bean-like substance that shields them from extreme pH. And also, should any penetrate, they have very efficient ion pumps to pump out any hydrogen ion that can clean, that is at, gets at acidic environments.
Amazing. All right, I forgot. Okay, well, let's give everyone a round of applause. That was an amazing talk. We have a DeSciNYC mug for you, and we also have a big science grading it hat for you as well. And you can keep the tardigrade for this. Fantastic! Yeah, thank you. All right, we have to go. So thank you guys all for coming. We have to clear out of the space, but thank you all, and we'll see you soon.
Thank you.